gram staining. Now, this gram staining procedure was established by scientist Christian Gram. Okay, what is the technique, student? Take a glass like this one, okay, and put a bacteria here. Okay, this is the culture of bacteria, and add here the staining. So first procedure is to add stain. Which stain you are adding, student? You are adding the basic dye. The name of the dye is crystal violet. Crystal violet, or it's called gram stain. Okay. Now with this, you are adding this stain. Then the bacteria, the color of the bacteria will be bluish purple. The color will be bluish purple. Then in the second step, add 0.5% iodine. Okay. Now earlier you have added crystal violet, that is CV. Then you have added iodine. Total complex is CVI complex, that is crystal violet iodine complex. The function of iodine that it acts as a fixant or mordant. They are adding, we are using it as an adhesive. It will help to adhere this staining to the bacterial cell, fixant or mordant. Then the third step, students, that de-staining. Okay, de-staining procedure. De-staining procedure means you will remove the stain with the help of alcohol. Okay. Now, what happens, student? Gram-positive bacteria have thicker cell wall. Okay, gram-positive bacteria have thicker cell wall, and gram-negative bacteria thin cell wall. Okay. Thin cell wall plus they have a layer of LPS, lipopolysaccharide. Okay. Now, due to this LPS layer, student, this gram-negative bacteria will de-stain the color. That means the bluish purple color, which was due to crystal violet, that will be de-stained. Why? Because due to thin cell wall and LPS layer. But this thicker cell wall, it will retain the color. So gram-positive bacteria will retain the color. Gram-negative bacteria will lose the color. So in the fourth step, we are adding counter stain. The name of the counter stain is safranin. With this safranin, gram-negative bacteria will be reddish in color or pinkish in color. Reddish in color and pinkish in color. Okay. Now, what is the difference with this gram-positive? Now, this staining procedure is also called differential staining. Differential staining. So let us see some differences with this gram-positive and gram-negative bacteria. Okay. Now, gram-positive bacteria, example of gram-positive bacteria, students, Bacillus. One more example we can take, Lactobacillus. This is also called lab. Gram-negative E. coli, agrobacterium. Remember, student, agrobacterium. In this bacterium, Ti plasmid is found. Then rhizobium. Vibrio cholera. Azotobacter. Now, these gram-positive bacteria, they are sensitive to penicillin. Sensitive to penicillin means they will be killed by penicillin. They will be killed by penicillin. Okay, why? Because this penicillin will prevent cell wall formation. That is peptidoglycan will prevent peptidoglycan formation. So the cell will die. Okay, the cell will die. But they can survive.
It prevents.
Yes, tell me, Agni.
Ma'am, it prevents formation of
Peptidoglycan. PDG means peptidoglycan. Okay, peptidoglycan formation.
yes, ma'am.
So they can survive in presence of penicillin. Because they has because of the layer of lipopolysaccharide. They have the layer of lipopolysaccharide. So if you want to kill this gram-negative bacteria, we have to use broad spectrum antibiotic. Okay. We have to use broad spectrum antibiotic. Broad spectrum antibiotic. In last lecture, I told you students that narrow spectrum, example of narrow spectrum antibiotic is penicillin and broad spectrum antibiotic is streptomycin. Streptomycin. Is this clear to you, student? So our next cell organelle is flagella. Now this flagella consists of basal body, hook, and filament. If you see the structure of flagella, students, I'm drawing the structure of flagella. Suppose this is the hook part. This is the hook part. Then they have a ring structure present, which is called L ring. Then another ring, P ring. Then another ring, S ring. Then another ring, M ring. So these are the 4 rings present in this bacteria. Okay. This L ring, it is actually in the layer of LPS, lipopolysaccharide layer. Okay. This P ring in the layer of peptidoglycan layer. This S ring in supramembrane ring. And this M ring, membrane ring. 4 types of rings are associated with the structure of flagella. And this region is filament. or this is also called shaft building. Okay. These are rod-like structures. Now this flagella protein, suppose this is flagella. Okay. Now this flagella consists of protein. In prokaryote, I am talking about prokaryote flagella. Prokaryote flagella consists of a protein called flagellin protein, whereas eukaryotic flagella consists of tubulin protein. Okay. The first two set, this LPS and PDB, these two set is called distal set. Distal set. They are present in cell wall region. And these two set, they are called proximal set. Okay. And they are present in the region of plasma membrane. So we can say, students, that in gram-positive bacteria, two rings are present. Okay. Two rings are present. What are that ring? S and M. Whereas in gram-negative bacteria, four rings are present. As you can see in the diagram. So this is a typical diagram of a gram-negative bacteria. Okay. Now this gram-positive bacteria, flagella is common. Okay. Flagella is less common in gram-positive bacteria, whereas flagella are more common in gram-negative bacteria. Structure of flagella is clear to you, students? Structure of flagella is clear to you, students? Clear to you, all of you?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
No. Any doubt, any one of you? Now, difference with prokaryotic flagella with eukaryotic flagella. Okay. Now, prokaryotic flagella consists of flagellin protein. As I told you, they are consist of tubulin protein. Okay. Next, they are monofibrillar, made up of only one fiber, monofibrillar. But here, you see the structure that is 9+2 arrangement, just like the structure of the centriole. These things you read in detail, just tell the unit of life chapter. Okay. Now, for their movement, ATP is not required. But here, for the movement of flagella, ATP is required. Okay. Now, here the type of movement is rotatory. Okay, rotatory means in 360 degree they will move. And here, in eukaryote, 180 degree, flapping movement. Like this, then like this. This type of movement is called flapping movement. Clear with the differences, students? Now, flagellar arrangements. What are the arrangements of flagella? Flagellar arrangements. If flagella is present, that type of flagella are called trichus. That type of bacteria is called trichus bacteria. Trichus means flagella present. Okay, A trichus. A means absence. Absence of flagella means flagella absent. Let us see the types. Suppose this is a bacteria. Flagella. One flagella is present. It is called monotrichus. It is called monotrichus. Okay. Bacteria, both sides single flagella is present. Then it is called amphitrichus. Amphi means both. Amphitrichus. This is a bacteria and tuft of flagella present on one end. That type of bacteria is called Cephalotricus. Okay. Tuft of flagella present on both ends like this. On both ends, they are called Lophotrichus. Lophotrichus. And suppose this is the bacteria and flagella present on all the ends like this, all the ends. then this type of bacteria is called Peritricus. Peritricus. Now, maximum study done on this Peritricus bacteria. Best example is E. coli. Now, here students, this one, from here to here, movement is polar. Polar means either they will move in this direction or in this direction. But here, in case of Peritricus, movement is non-polar. In all directions it will move, not according to pole. Okay. Is this clear, students? Students, is this clear? Clear to all of you?
Yes, ma'am.
Yes, ma'am.
Okay, next part is nutrition. Next part is nutrition. So nutrition basically on the basis of carbon source it will divide. So there are two types. One is autotrophs and other is heterotrophs. Now in heterotrophs they use organic carbon compounds. Okay, organic carbon compounds. In autotrophs they are using inorganic carbon compounds. Example, carbon dioxide. With the help of carbon dioxide, they fix energy and they prepare the food. So they are using light energy for photosynthesis. If they are using light energy, they are called photoautotrophs. And if they are using chemical energy, they are called chemoautotrophs. Yes, Chinmay, tell me. Tell me, Chinmay. Any doubt? Tell me. Chinmay, your voice is not clear. Do one thing. Chat me, Chinmay. Chat me, Chinmay.
Yes, ma'am.
Okay. Photoautotrophs are again of two types, students. Okay.
Ma'am, plants. Coscuta.
Your voice is not clear to me, Chinmay. Text me, Chinmay. Text me. Photoautotrophs. In photoautotrophs, again two types of student. Purple sulfur bacteria. Okay. And green sulfur bacteria. Now they absorb light, student. They absorb light. And here the pigment which is absorbing the light, that is bacteriopurpurin. Purpurin. Okay. And here that is bacterioviridae. The wavelength in which they are absorbing the light, that is for bacteriopurpurin we are using B. The wavelength of light is 890 nanometer. In bacterioviridae, that is B, 840 nanometer. Here the electron donor or electron or hydrogen donor is sulfurous compound. Sulfurous compound. Example, sodium thiosulfate. Okay. Example, sodium thiosulfate. And here we are utilizing water. To take one example, example is rhodospirillum. And here example is Florobium. What is the difference with the photosynthesis in bacteria with plants? That means bacterial photosynthesis to plant photosynthesis. What is the difference? In bacteria photosynthesis, one photosystem is present. That is bacteriobudding or bacterioperforating. But in plant, two photosystem is present. PS1 photosystem. Okay. PS1 and PS2. Okay. Here electron donor is other than water. Here electron and proton donor is water. In plant, donor is water. Here sulfur compound are the donor. That means water is not the donor. And in bacteria, photosynthesis takes place under anoxygenic condition. Anoxygenic means absence of oxygen. Okay. But in plant, oxygen requirement is compulsory. Okay. So here photosynthesis takes place in oxygenic condition. Oxygenic condition. This is the difference with the plant and bacterial photosynthesis. So let us see some example of chemoautotrophs. First example which I will put you here, that is nitrifying bacteria. Okay, nitrifying bacteria. What is nitrifying bacteria, student? Suppose ammonia. This ammonia is converted into nitrite. Nitrite. And this nitrite is converted into nitrate. That is why they are called nitrifying bacteria. Means they are able to convert ammonia into nitrate. Okay. Let us see some example. Nitrosomonas. Another example. Nitrococcus. Then nitrocystis. Then nitrobacter. These are the examples of nitrifying bacteria. Okay, students. The electrons which they will release here, these electron, student, the electron which they will release here, they will release the electron. They will release the electron. They will release the electron. And this electron will enter into electron transport system. And with the help of ATPase complex, they will synthesize ATP. Now this ATP will be utilized for carbon dioxide fixation. And they will prepare food with them. Okay. So this is nitrifying bacteria. Another category of bacteria is student iron bacteria. This here they are utilizing iron. Okay. Here they are utilizing iron. Second category is iron bacteria. So they are able to convert ferrous salts like Fe2 plus to ferric salt, that is Fe3 plus. Ferrous salt is converted into ferric salts. Take one example, students. Hydrogenomonas, the example of iron bacteria. Then third category we can take, that is sulfur bacteria. In sulfur bacteria, again two categories are there, student. See the categories. One is able to utilize, suppose hydrogen sulfur is converted into sulfur, means it is oxidizing. Another sulfur is reduced to sulfuric acid. Two categories. This category, under this category, we will get the example Iobacillus. And this category will get the example Vegia toa. Vegia toa. Okay. Now again, next category is heterotrophs. In heterotrophs, again, organoheterotrophs, photoorganoheterotrophs, and chemoorganoheterotrophs. Heterotrophs, photoorganoheterotrophs, chemoorganoheterotrophs. Photoorganoheterotrophs means here they are utilizing carbon source organic compound. Same here also. And electron source also organic compound. Electron source organic compound. Here also electron source is organic compound. Suppose when light energy is present. Okay. When light energy is present, suppose energy source light energy is present. Okay. So two categories of bacteria again here student. One is purple non-sulfur bacteria. They are sulfur bacteria we have read. They are non-sulfur, and here same also green non-sulfur bacteria. Again, photosystem are same student. Bacteriopurpurin. Here bacteriopurine. Now here electron sources. What is the electron source? Electron source is propyl alcohol. Here electron source is malic acid. Take some examples. Rhodospirillum, and here example chloroplexus. Second example is chlorolima. Rhodospirillum, and another example is
Chloro. First one is
Chloronema chloroplexus. Chloronema chloroplexus. Rhodospirillum, another example is Rhodopseudomonas. These are the example of purple non-sulfur bacteria and purple green non-sulfur bacteria. And chemoorganotrophs student. Chemoorganotrophs. Here how they are utilizing the energy? Oxidation of organic compounds. In this, you will get three categories, student. First one is parasitic. Second one is saprophytic. And third one is symbiote. Now, nutrition part is clear to all of you. Students, nutrition part is clear to all of you. Tell me, students. Nutrition part is clear to all of you.
yes, ma'am.
Yes, ma'am.
Okay. So our next category is reproduction. The reproduction, the basic we know, students, that reproduction are basically divided into categories, that is asexual reproduction and sexual reproduction. In asexual reproduction, only one parent is involved. They are uniparental condition. In sexual reproduction, biparental condition. Two parents are involved. Okay, here gametes are involved, and here absence of sex cells. So in asexual condition, you will see again two types, like binary fission. When they will go for binary fission? Under favorable condition. But when condition is not favorable, they will go for spore formation. What type of a spore, student? Endospore. Okay, in unfavorable condition. Unfavorable condition means suppose moisture is less, high temperature, high pH, low pH. These are the unfavorable conditions. Okay. And in sexual reproduction, what is the basic of sexual reproduction, student? Suppose this is male gamete. This is female gamete. They are haploid. Fusion takes place. And this is diploid condition. This condition is called zygote. Condition is called zygote. And this zygote will form new individual. How they form new individual? With genetic recombination. In bacteria, what happens, student? No gamete formation, no zygote formation. But genetic recombination takes place here. Genetic recombination takes place in bacteria also. This condition is called parasexuality. Why parasexuality? Because gamete formation absent, zygote formation absent, but they possess genetic recombination. This type of phenomena is called parasexuality. Okay. So our next category is student endospore. So what is this endospore? Endospore are nothing but they are hard, thick, resistant structure. Endospore. In unfavorable condition, they are forming. In unfavorable condition, they are forming. Okay. Suppose this is a bacterial cell, and in this bacterial cell, they are forming a hard, thick cell like this. Thick cell coating. This is thick cell. This thick walled structure formed inside bacterial cell are called endospore. Thick walled structure formed inside bacterial cell. They are called endospore. And nature-wise, they are endogenous in origin. Endogenous means they are forming inside the bacterial cell. That is why they are endogenous in origin. Okay. Now, when they will get favorable condition, what will happen? This thick wall will degenerate, and it will germinate into vegetative cell. Okay. What is the main function of this endospore, student? Suppose when endospore will release, in unfavorable condition, they will form thick walled structure. They are resistant structure. Resistant to what? Just now I am telling you, student. They are resting structure. But when they get favorable condition, students, so this thick wall will deteriorate, and they will germinate into two bacterial cells. They will germinate into new bacterial cell. What is its main function, student? Main function of endospore, they can survive in low temperature, around minus 100 degrees centigrade, and they can survive in high temperature, around 100 degrees centigrade. This is minus 100 degree, this is 100 degrees centigrade. So they are resistant to low temperature as well as high temperature. Okay. Now they have 4 layers. Endospore have 4 layers. What are the layers? Thick wall structure have 4 layers. One is exosporium. Second one is spore coat. Third one, cortex. Fourth one, core wall. Now students, see the normal bacteria can kill around 100 degrees centigrade. Okay, because in boiling water we have seen that all the bacteria are killed. Most of the bacteria are killed. So what special structure do they possess for which they can survive so much high temperature, around 100 degrees centigrade? That means if you are boiling water and that boiling water contains bacteria, endospore forming bacteria, like all bacteria has not ability to form endospore. Some bacteria has the ability. Okay. So that water contains endospore forming bacteria. So what will happen? You will give heat, that bacteria will not die. That bacteria will convert into spore. Which spore, student? Endospore. Okay. So there is something present in their structure which prevents them from heat. What is that structure, student? This name is very, very important, student. Okay. This name is very, very important. And its name is TP-O-linic acid. Okay. DPA. It is the anticoagulant it is anticoagulant. It is anticoagulant. Okay. This dipicolinic acid structure is present in the cytoplasm of endospore. Okay. Normally we know, suppose protein. Now that protein also denature at high temperature. I can give you one example, student. Denature. Denature means their protein structure will be destroyed. Suppose egg. Egg is protein. All of you know that egg is protein. You are giving heat to that egg. So what is happening, student? Egg undergoes the process of denaturation. That means its structure will destroy. And denaturation means now egg has been converted into omelette because you are giving heat to that egg. Okay, you are giving heat to that egg means that egg has been converted into omelette because that protein has undergone process called denaturation. So at higher temperature, protein denature. But in case of endospore, here protein is not denatured. Why, student? Denaturation or coagulation of protein doesn't take place. Why? Because here anticoagulant is present. The name of that anticoagulant is dipicolinic acid. Okay. That's why they can resist to higher temperature. So we can write here that denaturation of protein at high temperature is prevented by it. Denaturation or coagulation of protein at higher temperature is prevented by it. Because anticoagulant is present, dipicolinic acid. I hope its function and reason is clear to all of you. And one more thing, suppose they are not undergoing in any metabolic process, student. Endospore are resting structure. They are in a resting condition. So they are not undergoing into any metabolic activity. One more reason is water content is less in them. Okay, less, or we can say very less. Water content is very less. For any metabolic reaction, we know water is the main reagent. That's why metabolic activity also decreases and they undergo resting structure. Metabolic activity decreases. Okay, and due to this decreased metabolic activity, they prevent germination. They prevent germination during unfavorable condition. Unfavorable condition. Now here one thing you should know, student, the name pasteurization. Now, whenever you buy milk from outside, that mother dairy milk, okay, or Amul Taza milk, in that milk packet, have you seen in that milk packet it is written pasteurized milk. In that packet it is written pasteurized milk. So what is the meaning of this pasteurized milk? The meaning of this is that milk is heating. Milk is heated around 62.8 degrees centigrade. Milk is heated. Why they are heated? To kill bacteria. To kill bacteria. Now, what is the temperature? 62.8 degrees centigrade. Around 30 minutes you are heating the milk. And that milk is pasteurized milk. But you tell me, student, endospore forming bacteria can resist in 100 degrees centigrade. So does pasteurized milk is bacteria less or free of bacteria? No. Because endospore forming bacteria are still present inside that milk. Because endospore they are resistant. So we can say that pasteurization, these endospore forming bacteria are resistant to pasteurization. Resistant to it. These endospore forming bacteria are resistant to pasteurization. In pasteurized milk, there is a possibility to get endospore forming bacteria. Till this clear to you, all of you? Till this clear, students? Till this clear,
Yes,
students,
ma'am.
all of you?
Yes, ma'am.
Endospore portion is totally clear now? Okay. Next is parasexuality. Parasexuality, that is genetic recombination in bacteria. Okay. Parasexuality, genetic recombination, transfer of genetic material. Now, first we will read, student, transformation. Scientist name proposed, that is Griffith. Bacteria he used Diplococcus. Streptococcus. Pneumococcus. What is the definition, student? Picking up of foreign DNA from surrounding. Second phenomenon we will read conjugation. Experiment given by Lederberg and Tatum. Bacteria used E. coli. Okay. Now, transfer of genetic material with the help of
Ma'am, Lederberg and
yes, tell me. Lederberg and Tatum. Lederberg and Tatum. Transfer of genetic material with the help of conjugation tube. Conjugation tube. And third experiment is translation. This was given by Lederberg and Zinder. We used Salmonella bacteria name. Salmonella. Okay. And here virus particles are released. With the help of this method, virus particles are released. Now, the first process, student, that is transformation. Okay, transformation. Now, Diplococcus, bacteria Diplococcus, it consists of two types of strain. Okay, one is R strain and another is S strain. Why two types of strain is there, student? Let us see the Diplococcus. Diplococcus is the bacteria. Two types of strain. One is R strain, another is S strain. Now, R strain means capsule is absent, student. No capsule layer is present. That's why it gives rough appearance, rough colonies. Okay, rough colonies. And they are non-pathogenic. They are non-pathogenic. Because capsule layer is absent, but here capsule layer is present, which will give the pathogenicity to the bacteria. And due to this capsule layer, they are producing smooth colonies. That's why S and R name has been given. And they are pathogenic. Now, what is the experiment, student? Let us see the experiment. What he did, he means I'm talking about Griffith. Okay. He give living R type. Okay. He give living R type. He injected into mice. And this mice survived. Next time he has done what? He gave living S type. S type means pathogenic type. So this time mice killed. Second type. Third type, what he did, he gave heat killed S bacteria. Heat killed means we are killing the bacteria by giving heat. Heat killed S type. Heat killed S type. Heat killed S type means you are injected in mice. This time mice survive. Why? Because we are giving heat. Heat is killing the bacteria. These three are the general phenomena. No issue. Now, main thing is the fourth step. That is, you are giving heat killed S type. And you are giving living R type. Living R type means bacteria should survive because living R type. Heat killed means bacteria should die. Sorry, heat killed means bacteria will survive. And living R type means bacteria will survive. But in this condition, mice died. Now, it was confusing. Why mice died? Because we have given the heat kill type. That means we have already killing the bacteria. Then how they are surviving? And R type doesn't possess any capability to kill that mice. The reason is that this heat killed S type is producing some chemical which is converting this R type into H type. And due to this living H type, the mice actually dies. So he said that some transforming principles are there. Griffith told that some transforming principle is present, which is responsible for the conversion of R to S type. This was the conclusion of Griffith experiment. But he didn't mention what is that transforming principle. Today we know that that transforming principle was DNA. But at that time Griffith didn't know it because he was doing the experiment. Okay. Now after this Griffith experiment, three scientists after this Griffith experiment, the chemical nature of this transforming principle, chemical nature of this transforming principle was discovered by three scientists. Very important student, the name of the scientist. First one is Macarism. Second one, McCarthy. Third one is Avery. They told that this chemical nature, that genetic material is DNA. That chemical nature is the genetic material, and that genetic material is DNA. Now see the mechanism, students. Suppose this is R-type bacteria. Okay. This is C minus. C minus means capsule is absent. Okay. But this R-type bacteria has the capability to take DNA. So this type of bacteria is called competent. Competent means the bacteria which has the ability to take DNA is called competent. This phenomena is called competence. The phenomena is called competence. So this R-type bacteria has the ability. Okay. Now this R-type bacteria has been converted into S-type bacteria. Why? Because this C minus has been converted into C plus. That is, capsule is present. With that heat-killed bacteria, that capsule part, that genetic material actually enter into this R-type and converted this R-type into S-type. Now, if it is converted into S-type, they have the ability to produce capsule surrounding their body. And when I taught you capsule, student, remember, capsule gives pathogenicity to bacteria. And not all bacteria has the probability to form capsule. But capsulated bacteria, they are also not engulfed by WBC in our body. Normal bacteria, generally, they are killed by WBC. But if any capsulated bacteria has entered into your body and it causes infection, student, then that bacteria will not get engulfed by WBC because of the layer of capsule. So we can say now that capsule give defense to the bacteria. Capsule is responsible for the pathogenicity to bacteria and gives additional benefit to make them survive. This capsule layer. Clear with the experiment, students? This Griffith experiment is clear to all of you? Students? Griffith experiment clear to all of you? Yes, Agni, tell me. Clear to all of you? Griffith experiment?
Yes, ma'am.
Yes, ma'am.
So, what we have done today, student, just a quick topic revision. Firstly, we have seen the gram staining process. Okay. We have seen the differences with the gram positive and gram negative bacteria. Then we have seen the structure of flagella. Okay. And what is the difference with the gram positive and gram negative bacteria with respect to flagella? Okay. Then I have discussed, student, types of flagellar arrangement. Okay. Flagella are absent, flagella are present, monotrichous, amphitrichous, cephalotricus, lophotrichus. Then I have discussed the nutrition part, student, like photoautotrophs, heterotrophs, chemoautotrophs, that nitrifying bacteria, iron bacteria, sulfur bacteria. Okay. Then the reproduction part, that is both asexual and sexual reproduction. Then we have seen the detailed discussion of endospore, students. Okay. Then pasteurization, and last of all, we have seen the Griffith experiment. In the transfer of genetic material, three methods are there: transformation, conjugation, and transduction, student. Okay. This is actually transduction. So first method we have seen transformation today. This is transduction. Okay. First method we have seen, that is transformation, conjugation, and transduction. I'll discuss in next lecture, students. Clear with this, all of you? Students, clear with this, all of you?
Yes, ma'am.
Okay. With this, I am ending today's lecture. Okay. Thank you all.